All right, on this episode of Python Papi, we're still practicing with the princess. Right now, I just want to see how far her knowledge goes and how many subjects she can actually touch on. So let's do that now. Princess, what time is it?
The time is currently 11:02 a.m.
Princess, tell me a joke.
There are 10 types of people, those who understand hexadecimal and 15 others.
Princess, tell me about gene mapping.
Gene mapping or genome mapping describes the methods used to identify the location of a gene on a chromosome and the distances between genes. Gene mapping can also describe the distances between different sites within a gene. The essence of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. Molecular markers come in all forms. Genes can be viewed as one special type of genetic markers in the construction of genome maps, and mapped the same way as any other markers. In some areas of study, gene mapping contributes to the creation of new recombinants within an organism. Gene maps help describe the spatial arrangement of genes on a chromosome.
Princess, tell me about DNA sequencing.
DNA sequencing is the process of determining the nucleic acid sequence, the order of nucleotides in DNA. It includes any method or technology that is used to determine the order of the four bases: adenine, guanine, cytosine, and thymine. The advent of rapid DNA sequencing methods has greatly accelerated biological and medical research and discovery. Knowledge of DNA sequences has become indispensable for basic biological research, DNA genographic projects, and in numerous applied fields such as medical diagnosis, biotechnology, forensic biology, virology, and biological systematics. Comparing healthy and mutated DNA sequences can diagnose different diseases including various cancers, characterize antibody repertoire, and can be used to guide patient treatment. Having a quick way to sequence DNA allows for faster and more individualized medical care to be administered, and for more organisms to be identified and cataloged. The rapid speed of sequencing attained with modern DNA sequencing technology has been instrumental in the sequencing of complete DNA sequences, or genomes, of numerous types and species of life, including the human genome and other complete DNA sequences of many animal, plant, and microbial species. The first DNA sequences were obtained in the early 1970s by academic researchers using laborious methods based on two-dimensional chromatography. Following the development of fluorescence-based sequencing methods with a DNA sequencer, DNA sequencing has become easier and orders of magnitude faster.
Princess, tell me about Margaret Sanger.
Margaret Higgins Sanger, born Margaret Louise Higgins, September 14, 1879 September 6, 1966, also known as Margaret Sanger Slee, was an American birth control activist, sex educator, writer, and nurse. Sanger popularized the term birth control, opened the first birth control clinic in the United States, and established organizations that evolved into the Planned Parenthood Federation of America. Sanger used her writings and speeches primarily to promote her way of thinking. She was prosecuted for her book Family Limitation under the Comstock Act in 1914. She feared the consequences of her writings, so she fled to Britain until public opinion had quieted. Sanger's efforts contributed to several judicial cases that helped legalize contraception in the United States. Due to her connection with Planned Parenthood, Sanger is frequently criticized by opponents of abortion.
Princess, go to sleep.
Until next time, Mr. Gates.
All right, like I said, that was just some practice I was going over with the princess, just to see how many topics she could touch on without an issue. So yeah, we're gonna keep on feeding her, making her more stronger, making her more robust, and stay tuned. This is the Python Poppy. You guys stay Gucci.